Now we are ready to show the continuous spectrum experiment itself. As it is shown in the book, we have a flashlight here, and this is shining the light onto the grating. We have, of course, again gratings of different grating constant. We have put on one of them, and because this flashlight is an incandescent light and it produces a continuous spectrum, here the electrons jump up and down without any correlation to the photons that come out of this electron jump. So you can easily see that this is a spectrum. One can see that here the entire spectrum is as if the colors are smeared. You can see practically the red here, the orange, the yellow, the green, the blue, and a little bit of violet. So human eye can see almost up to the edge of violet, about 350 nanometers, and it can see up to about 750 nanometers on the red side. The important thing to notice is that there are no discrete lines here. The whole thing is a continuous spectrum. And what you can do as far as the experiment goes is to mark the colors with your pen and see how many colors you can distinguish, and go back and look at the chart to find out the wavelengths of these particular colors, remembering that each color consists of a whole bunch of waves. Now we are ready to see the spectra themselves. We have described these three different types of spectra. This is the continuous spectrum. We have got red on one side and the violet on the other side. The red continues to orange, and that continues to yellow, green, blue, and violet. All the 7 colors are here, but there's no distinct lines in this kind of spectrum. That's why it's called a continuous spectrum. Incandescent light will give this kind of spectrum. This is where the electrons jump from the ground state to different levels at different time intervals, and there's no phase correlation or wavelength correlation between the photons that are given. They're completely at random. Such a thing gives you this spectrum, which is what we have seen in our experiment. The second type of spectra are the emission spectra. Notice that there are bright lines here on a dark background. You can see them. If, for example, in the barium you see a large number of colors, whereas if you go down all the way down to sodium, for instance, this has got only one yellow line. So these spectra are specific to the elements which are excited, which means that these electrons from the ground state will not go at random anywhere, but to only to specific levels. From there they jump and give only those wavelengths. In other words, you can say that the emission spectrum is a fingerprint of that element. So as a matter of fact, you can identify the element by looking at its spectrum. This is extensively used in spectrochemical analysis. The last one we have got is the absorption spectrum. In this one, the spectra look very similar to the continuous spectrum, except a bunch of dark lines. So this kind of spectra are produced when bright light coming from a hot surface, when it goes through cold gas, the gas absorbs certain wavelengths and let go the rest of the light. That's why this absorption spectrum looks like a continuous spectrum, and then with number of black lines. This particular spectrum, which you are at the top, is the solar spectrum. This is a very important spectrum because we can say that this spectrum is the starting of the quantum mechanics, because at that time it was very not possible to explain these Balmer series, Paschen series, and so on and so forth. Only after Bohr talked about the stable orbits, we are able to explain where these spectra come from. So therefore, these are the 3 types of spectra. In our experiment, we have seen the continuous spectra. Now we will be describing the emission spectrum, also the spectrum experiment itself. Then you can correlate between these two. The experiment itself consists of first looking at the emission spectrum and determine the wavelengths, observing the wavelengths through the spectrograph and comparing with this spectra, and then determine the energy and so on and so forth. Now we will see the actual experiment for observing the emission spectra and making a calculation for the energies. Here is a holder in which you have a gas, excited gas in a tube, and this can be excited by a power supply, and this gas will be illuminating, and this light is taken through the spectrograph. This is the spectrograph. In this one there is a slit here which is adjustable, which I will be showing you in a second. Light comes here, and this is the dispersal element, which is the grating, and the light, in order to increase the dispersion, is taken to another place, and where you have got a mirror, this will be reflecting it back, and it goes to the eyepiece, and you can observe the spectrum here. So when you properly adjust it such that this light is right in front of the slit, you can see the spectrum from here. There is also, in the same focal plane as the spectrum, there is a wavelength scale also. So if you put this one here, you can illuminate the wavelength scale. This is just an incandescent bulb. This will illuminate the wavelength scale, and the wavelength scale will be superposed on the spectrum itself. So therefore, we can see the spectrum, and you can observe the wavelengths and make a note of the wavelengths of the different lines. You can replace this by different gases. You have got hydrogen, you have got mercury, and so on and so forth. 4, 5. These tubes are very inexpensive, and this whole setup is very inexpensive. You can do it in any lab. I will now turn this one to show you some important things. This is the slit. This slit is adjustable. There is a screw here. Initially, you can use a wide slit, and then when you are ready, you can reduce the slit width so that the lines will be very sharp. And then you have got this eyepiece through which you will observe the spectrum, and you just put your eye here and you can see the spectrum. And because this incandescent lamp lights the wavelength scale, you can see them simultaneously. The only important thing when he is here, that the slit and the excited tube must be fairly well aligned so that you can see the, well, you can actually move the spectrograph a little bit here and there to see the spectrum very well. Once you see the spectrum, you will note down the various wavelengths, and from the wavelengths you will compare it with the chart. And because you know the wavelength, and you know the velocity of light, and you know the Planck's constant, you can calculate the energy difference. The energy difference, of course, comes out in joules, but the joules you can convert in electron volts by actually using the 1 electron volt equals to 1.6 times 10 to the power of -19 joules. So this is how you would fill up that chart.